Thank you for watching today's presentation entitled Predictive Cancer Models Using Patient-Derived Xenograft Mice. My name is Janine Lohmar-Kelly. I am delighted to share this educational information to support you in achieving research excellence. If you have questions about the content of this presentation, please contact us using the information found on the final slide of this presentation. To begin, let me introduce you to the Jackson Laboratory. The Jackson Laboratory's mission is to discover precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. We fulfill our mission as follows. We have our own research staff who investigate mechanisms of human disease partly through the use of the laboratory mouse. We also provide scientific services and resources to investigators around the world. This includes mice such as the NSG model as well as patient-derived xenografts, which we will discuss today. Last, we are committed to education and outreach. In addition to this educational recording, we also sponsor traditional conferences and hands-on workshops. We educate students of all ages and experience levels through courses, internships, and postdoctoral training. With regards to our services and resources, the Jackson Laboratory is the world's largest repository for mouse models of human disease. We are funded through the National Institutes of Health to preserve as many mouse strains as possible to ensure their availability for all time. Among the more than 11,000 mouse strains that we distribute, over 1,500 strains are maintained as live colonies, and relevant to today's presentation, about 700 strains have been annotated as cancer models. JAX mice are the best characterized and referenced mice in published research. Much of this can be attributed to our unsurpassed genetic quality and rigorous animal health standards. I highlight the health standards today because we are discussing the highly immunodeficient strain NSG. It is the use of our well-characterized and heavily referenced common inbred strains such as the C57 Black 6J or BALB CJ mice which supports the collection and distribution of specialty mutant mouse models that cannot be found elsewhere. In addition, we have the following freely available resources. The JAX mice database serves as a centralized catalog for those 11,000 strains I mentioned before. In addition to practical information like genotyping protocols and breeding information, we list phenotype information using a defined vocabulary and link the relevant publications from which the data was collected. The Mouse Genome Informatics database includes information not only about mice from JAX, but also from mice from other repositories and other investigators. The Mouse Phenome database is a collaborative collection of baseline phenotype data for some of the more popularly used strains. And finally, two relevant resources for today's content, the JAX Clinical Knowledge Base, which may be helpful for oncologists to decide treatments based on the genetics of the tumors, and finally, the Mouse Tumor Biology Database, which has tumor information in mouse strains and also houses our patient-derived xenograft data. Again, all of these are free online resources available to you to help with your research needs. So with regards to this presentation, these are the learning goals. By the end of this presentation, you should be able to describe which components of the NSG mouse make it useful as a platform for human disease research, particularly for the use of patient-derived tumors and immune cells. You should also be able to demonstrate how PDX models are used for predictive and preclinical research applications. And last, you'll be able to combine PDX and human immune cells into a sophisticated immuno-oncology model. So let's begin. You may realize that the biggest obstacle for cancer treatments is getting through preclinical testing. For oncology compounds entering into phase 1 clinical trials, only about 5% become approved for use in patients. Most compounds leave the drug development pipeline due to efficacy and safety concerns in humans. One of the reasons that this may be happening is a limitation common to all animal models to accurately recapitulate the complexity of human cancers. Models are either genetically or experimentally induced mouse cancers, or they are human cancers or cancer cell lines that are xenografted into mice. Many of these models on their own fail to represent the enormous genetic diversity of human tumors and the genetic diversity of human patients. So to gain a better translational applicability to the clinic, there is a crucial need to use patient-derived xenografts, or PDX, as an additional model to the classical models to begin to bridge this gap. Historically, the classic xenograft model discovered in the 1960s was the nude mouse shown here. In this particular experiment, nude mice were injected subcutaneously with the human MCF7 breast cancer cell line. This is a human breast cancer cell line that has been propagated in tissue culture for many generations. These mice were treated with the experimental chemotherapeutic, in this case marodantin, intramuscularly every other day for 40 days. And you'll see on the left is the vehicle-treated mice, and on the right is the marodantin-treated mice. You can appreciate, because the mice are hairless, it is quite easy to see and measure the nodule with calipers. However, one major limitation to this model is that nude mice can only engraft cell lines and not primary patient tissues like a PDX. Cell lines are clonal populations of one type of cell and therefore do not have the diversity of cell types that can be found in real tumors. Therefore, nude mice and cell line work are generally very good for mechanistic studies, but may be poor at predicting clinical response. The challenge in studying human tissues in a mouse is that the mouse has an immune system that functions like a barrier to foreign cells. And I'm representing that immune system very simply on this slide. It can be broken down into innate and adaptive immunity, and many of the components here are important barriers to engraftment of human tissues. So therefore, to engraft tissues into a mouse, the mouse first must have some kind of defect in its immunity. Mice with the spontaneous nude mutation in the FOXN1 gene lack a thymus and therefore lack mature T cells. Because T cells interact with B cells, some B cell functions are limited as well. This lack of T cells allows engraftment of either mouse or human cancer cell lines, which are very hardy cells and grow very well both in culture and in vivo in mice. So we can summarize that the barrier in nude mice is generally too high for some more sensitive cells, such as primary patient cells, but the barrier is quite low enough for very hardy cells like cell lines. One solution to this problem of not being able to engraft primary human cells is to create a mouse with a very low barrier to engraftment. Another way to think of this is to use a mouse that is highly immunodeficient, such as the NSG or NOD-SCID gamma mouse. So we'll go through each of these components here. The N in NSG stands for NOD, or non-obese diabetic mouse. The NOD is a mouse strain that, when compared to other mouse strains of other genetic backgrounds, has a relatively lower immune function. For example, NOD mice have no complement. Compared to other mouse strains, NOD dendritic cells, natural killer cells, and macrophages are less functional. And last, they have a more human-like SERP-alpha allele on their macrophages, which allows for better engraftment of human hematopoietic cells, which can be used to create humanized immune systems, which we'll discuss later. The S in NSG stands for the SCID mutation in the gene PRKDC. This gene is important for repairing breaks in the double-stranded DNA. Because T and B cells must break, rearrange, and then repair DNA to create T cell receptors and B cell receptors, mice with a SCID mutation lack mature T and B cells. And finally, the G in NSG stands for gamma, or the interleukin-2 receptor gamma chain loss of function mutation. This gamma chain in this receptor is important for signaling for 6 interleukin cytokines. In particular, IL-15 signaling is lost, blocking natural killer cell development. Now when we represent NSG on a similar immunity diagram as before, you can appreciate that the barriers to human cell engraftment are quite low, as shown by the fading of all of these immune components into the background. You'll notice that NSG mice do have granulocytes and some macrophage function. However, overall, this mouse is very, very immunodeficient compared to the nude mouse. The NSG mice is highly immune efficient, which allows engraftment of sensitive primary human cells like patient-derived xenografts and human hematopoietic stem cells. Compared to other genetic backgrounds, in the NSG, there is no SCID leakiness in these mice, which is to say that the T and B cells remain absent. In other genetic backgrounds, there is a SCID leakiness in which these T and B cells may come back. In addition, compared to the non-SCID mouse, which has the lack of the gamma mutation, the NSG lives longer due to a delay of onset of thymic lymphomas that are common in non-SCID mice. And so NSG mice can live up to greater than 16 months without the impact of thymic lymphoma. And then finally, because of the specific SERP-alpha allele from the non-genetic background, the NSG mice also engraft human hematopoietic stem cells and could be useful in some immuno-oncology applications. However, because of their profound immunodeficiency, NSG require aseptic housing and handling to remain healthy. Without this aseptic handling and housing, NSG could get very sick from infections that more immunocompetent mice like the nude mouse would not have. And last, because of the SCID mutation and their inability to manage double-stranded DNA breaks, the NSG mouse tends to be more sensitive to irradiation and chemotherapeutics. However, as you'll see in the future slides, NSG mice can be treated with some of these DNA breaking and damaging agents. So the profound immunodeficiency of the NSG mouse now allows engraftment of primary patient tumor samples. At JAX, we receive tumors directly from the operating room. Within 24 hours of resection, we implant the fragments subcutaneously in the flank of an NSG mouse. All clinical information about the tumor type and drug treatment history comes directly from the clinicians that are providing the samples. These tumors that are engrafted are named P0 or passage 0. We then further expand the tumor into serially engrafted NSG mice, which we then call the P1 mice. At both of these passages, P0 and P1, we collect histology sections and perform immunohistochemistry, gene expression analysis in the form of RNA sequencing, copy number variation, and other analyses. We cryopreserve early passage tumor fragments, never culturing or dissociating the tumors into individual cells. This lets us retain the maximum genetic and phenotypic heterogeneity that is seen in patients. Some of our quality control and standard characterization that is done by our PDX resource includes short tandem repeat or STR testing to ensure that we are engrafting and expanding human tissues and to match them to their original patient samples. We also stain for human CD45 and mouse CD45 so we can test for the growth of lymphoma, which is a common phenomenon in engrafting human tissues into immunodeficient mice. And finally, we do some standard of care dosing to show how the tumors grow as well as how they may handle against common therapeutics found in the clinic. The cryopreserved tumor fragments can further be implanted and expanded into NSG mice that we call donor mice or P2 mice. The donors can then grow enough tumors to be implanted into what would become the experimental cohorts or P3 mice, which may be used for efficacy testing. The histology, treatment response, and growth rates suggest that the tumor fragments retain a high level of heterogeneity throughout low passages. However, it is a general feature of all PDX that over several passages of tumors in the mice, that heterogeneity of the tumor cells and of tumor-associated cells will be lost. The ability to answer questions about the stroma and about the patient's infiltrating leukocyte populations, therefore, will be limited. As an example, here are some of the sources of where we receive our patient-derived xenograft tumors. As you can see, these are collaborations that we have with 25 distinguished cancer hospitals around the United States, including Maine, California, Utah, Texas, and Florida, which all have very distinct geographical locations and distinct populations that they serve. Please note we're also constantly getting new tumors and adding to this collection. Currently, there are over 400 models in our collection. And you can see here that most of these are lung and colorectal tumors. Initially, when we started this program, all of the patient-derived xenografts were an all-comers tumor bank in which we did not distinguish between what types of tumors that were coming, and we tried to engraft as many as we could. As of late 2017, we have started collecting more triple-negative breast cancers, more drug-resistant tumors, and more rare tumors. Now that I have described the mice and the PDX collection in general, what does it look like when you put these two components together? Well, one very important feature to study PDX is whether the mouse model can retain the original patient tissue structure as well as their markers. In this particular experiment, NSG mice and at the time the BALB/C SCID mouse, which was the best mouse model of the time for immunodeficiency, were compared side by side and engrafted with similar pieces of the patient lung tumor. I hope you can appreciate that the tumors in the NSG mouse appear histologically more like the original patient sections, and the stains for human-specific HLA antigen. In contrast, the tumors engrafted in the CB17 SCID mice had a more fibrotic appearance and exhibited greater infiltration of host immune cells. After 9 weeks of growth in the NSG mouse, that same tumor retained a more typical appearance similar to the original patient sample by both structural H&E staining as well as for cytokeratin staining, which is a component of epithelial cells and a marker for carcinomas. Finally, as shown by staining for the proliferation marker Ki67, the engrafted tumor in NSG mouse retains its neoplastic potential. Together, these two slides support the idea that the severe reduction in host immunity of NSG mice permits a more faithful remodeling of the human tumor. These are two more examples of how the PDX replicates human structure. Here in the top panel is a colon adenocarcinoma with a KRAS G13D mutation implanted into the NSG mouse. You can appreciate that there is a retention of these crypt-like structures in the P0 and P1 mice. And the bottom panel shows a different colon adenocarcinoma that has metastasized to the lung. While this particular PDX was not analyzed in the mouse lung as it was implanted in the flank, you can see the similar structures in both the patient as well as the P0 mouse model. Once the tumors have been successfully engrafted into mice, it is important to note that the growth characteristics and potential to respond to standard of care treatment can be replicated. In this example, this bladder tumor was engrafted and grew a small nodule. Early in that growth period, the tumor was treated with cisplatin, shown in red, or saline, shown in black. As you can see, the cisplatin treatment delayed the tumor growth quite significantly, supporting the theme that PDX models grown in NSG mice may show a similar response as a tumor would have in a human patient. Something you may notice here is that the error bars in these experiments are quite large. As I mentioned before, one of the strengths of using PDX is the heterogeneity of the tissue in modeling clinical disease, especially compared to clonal cell lines. However, with this strength comes an experimental consideration that a larger sample size may be needed to produce robust and well-powered studies, which I'll show you in the next slide. This is the same data as before, but now just showing individual tumor-bearing NSG mice and only of the cisplatin treatment. You'll see here that each line is a single tumor-bearing mouse, and each dot is the tumor dimension measurement that was taken roughly 2 times per week. In red, you can see 2 mice with tumors that were resistant to cisplatin. And in blue, you have 1 mouse with a tumor that had extreme sensitivity to the drug. And everywhere else in black in between are mice with an intermediate response. So if you're used to seeing efficacy data using a cancer cell line, this heterogeneity shown here can be quite intimidating. However, this heterogeneity can be seen as a strength of the model, as it could be used as a tool to understand mechanisms of drug resistance. In addition, this is reflective of the heterogeneity of human tumors to respond differentially to a same drug. Shown here is an example of a different bladder tumor in response to the same cisplatin treatment. Generally, you might notice between these two models that there are differences in growth rate and differences in response to the drug. While PDX are more heterogeneous than clonal cell lines, it is important to consider that one PDX model is still only from one patient. So to better represent the heterogeneity of human patient populations to a drug response, you may actually want to consider using more than one tumor model per cancer type. You can take this idea quite far and use PDX as a platform for miniature clinical trials. So shown here is an experimental design using 5 non-small cell lung cancer tumors with differing genetic profiles, particularly in their EGFR status. One advantage to this kind of study is that each PDX can serve as its own internal control and can be compared to several experimental drugs or dosing regimens, shown here as A, B, and C. So this is a study design that works in mouse models but could never be done in a clinical trial because you cannot test out multiple types of dosing regimens on an individual patient, but you can with individual mice with the same PDX model. I'll now highlight another significant achievement of our PDX models particular to NSG mice. These are models in which we can grow ER-dependent breast tumors or estrogen receptor-dependent breast tumors. These are tumors that require estradiol supplementation for growth. From an experimental perspective, an unfortunate side effect of estradiol supplementation is the formation of estradiol crystals in the mouse's bladder. However, NSG are relatively more resistant to estradiol-induced bladder toxicity than other host strains and therefore affords the ability for ER-positive tumors to engraft. And you'll see here in this diagram that the patient model here, as well as the P0 and P1 passages, retain that same characteristic growth pattern. You'll notice as well that the estradiol supplementation allows the maintenance of ER positivity, as shown here by immunohistochemical staining. We'll also show in this particular model, which was also progesterone receptor positive, the PR positivity was also retained, as well as shown in the immunohistochemical staining on the right. So this is a unique feature of using NSG mice to be able to study ER-positive tumors in a mouse. Here's a new example. This is a human non-small cell lung adenocarcinoma, and the following slides will demonstrate the importance of molecular characterization and how it can be used to develop tumor type-specific therapies. This tumor retained its histological fidelity through passage 2, shown in these models here. But importantly, that we'd like to highlight is this tumor carries a mutant KRAS G12C, a wild-type EGFR, and has low expression of a third marker called ERCC1. Based on this gene expression profile, the prediction is that the tumor would be resistant to EGFR targeting drugs erlotinib, which target mutant EGFR, which is constitutively active, and sensitive to cisplatin because of the ERCC1 expression. Experimentally, when these PDXs were put into mice and treated with the same drugs, the gene expression does indeed predict the drug susceptibility. So the tumor here is resistant to EGFR. As you can see in the black line, the vehicle control, the tumor is growing. And when the tumor and mouse are treated with erlotinib, the tumor continues to grow. However, when treated with cisplatin, as predicted by the ERCC1 expression, the tumor growth is inhibited. So this experiment validates the utility of the NSG PDX model and the gene expression profile inherent in the PDX model to accurately predict drug response in human patients. This is another example in which the dosing is quite a bit more complex, and you can see how the patient was treated over time and how the PDX model in mouse will reflect that similar treatment scheme. So here we show a timeline along the x-axis of a patient's diagnosis and along the y-axis the tumor burden. This is a patient that had a somatic mutation in EGFR. When she was first diagnosed, the patient did not respond to first-line chemotherapy, shown here. The patient was then given erlotinib, and the tumor began to regress. And months later, the tumor developed a resistance to erlotinib and then rebounded. And at this point, the tumor fragment was collected and established in a PDX model in an NSG mouse. The patient then continued on treatment and eventually responded to an afatinib and cetuximab combination therapy. So using P3-PDX from this patient, efficacy studies were performed using the same drugs that were used in the patient's treatment. Remarkably, the tumor was resistant to erlotinib, similar to what happened in the patient, where there's this initial response of erlotinib, but eventually the tumor grows out. The tumor was completely blocked with cetuximab alone or in combination with afatinib, which was similar to what happened in the patient's response and treatment. This experiment demonstrates a possibility for the mouse PDX model to offer a personalized medicine approach. A PDX mouse platform could be used to try multiple drug combinations to help inform clinicians of potentially efficacious treatments. Putting these last two examples into a broader context, perhaps by combining PDX gene expression data with experimental efficacy data, we may be able to predict which treatments will be most efficacious based on a patient's individual tumor expression profile. As I mentioned earlier, the highly immunodeficient NSG model could engraft PDX as well as human hematopoietic stem cells due to the presence of the more human-like SERP-alpha allele. It's this characteristic of the model that has allowed us at JAX to create a sophisticated mouse that has been primarily used to test immunomodulatory therapies in which the drug targets are human immune cell markers. In many cases, these are immune checkpoint inhibitors. For these particular experiments, NSG mice are irradiated to clear the bone marrow niche and then injected with human CD34 hematopoietic stem cells taken from cord blood donations. After several weeks, the humanized immune system develops in these mice. At this time, the mice are then engrafted with either a patient-derived xenograft or a cancer cell line. And together, this creates a tumor-bearing NSG mouse with a humanized immune system, which we call OncoHu. When creating these mice initially, it was unknown whether the PDX would grow in the presence of an immune system that came from a different patient donor. In other words, the HLA types would not match, and it is possible that the PDX would be rejected by the different immune system. For the 3 experiments listed here, PDX that had already been known to grow in NSG mice alone were engrafted into CD34 humanized NSG mice. We did not attempt to match the HLA between the CD34 donor and the PDX donor. Surprisingly, there was no strong difference in PDX growth kinetics between the CD34 humanized NSG mice shown in pink and the non-humanized NSG mice shown in green. In our hands, most, but not all PDX models that we have already established to grow in NSG mice will grow in the presence of an unmatched CD34-derived humanized immune system. However, every PDX and every CD34 donor combination will vary from one experiment to the next, and there are some combinations in which the PDX may not grow. Currently, it is unknown which combinations of donor PDX and donor CD34 will be compatible and which will not be. For this reason, it is recommended that several CD34 donors be used in each experiment, evenly distributed in the experimental groups, as a way to protect against losing an entire experiment due to incompatibility between donors. Now that we can show that the PDX can grow in the presence of an unmatched HLA humanized immune system, we next validated that the models do respond to standard of care therapies. In this particular experiment, we first validated that the OncoHu mice could be treated with cisplatin, shown here in green. Similar to previous PDX validation studies, tumors responded to this common type of chemotherapeutic. For immuno-oncology applications, OncoHu mice were then treated with Keytruda in pink, a checkpoint inhibitor used clinically. Keytruda binds and blocks PD-1 on T cells and prevents its interaction with PD-L1 on tumor cells. This binding removes the tumor's inhibitory signal on the T cells and allows those T cells to now respond against the tumor, resulting in a reduction in tumor volume. Because the PDX and CD34 come from different donors, the mechanism for this tumor reduction is likely an allogeneic response. However, this study still demonstrates that the OncoHu model can be used to test the efficacy of immunomodulatory therapeutics and allow the immune system to recognize and eliminate these disease cells. Now that I've given you several examples of questions that can be answered using patient-derived xenograft models, I can now show you how to access the models from JAX. From the MGI resource that I described earlier, you can click on Mouse Models of Human Cancer. This opens the mouse tumor biology database where you can learn about the mouse tumors and tumor incidence rates. In addition, in the left menu under Additional Resources, click on the PDX Model Search link. This links you to the roughly 400 PDX models we have established, characterized, and distribute at JAX. The Patient-Derived Xenograft search form has been designed to allow searching for PDX models by several criteria. You can search by the primary cancer site here. This search can be further limited to only return results from models with available dosing data and from treatment-naive patients. You can also search by diagnosis based on the tumor characteristics such as adenocarcinoma, sarcoma, etc. You can also search by variants, searching for tumors that express specific gene variants such as KRAS G12D or KRAS G12V, etc. You can also search by gene expression, including oncogenes and tumor suppressors and other genes of interest. Please note that not all targets are listed in this database, but more RNA sequence data exists for all of these models in which you can contact us directly to learn about. Finally, there's also copy number variation in which you can search for tumors which the dosage of a particular gene is either amplified due to a duplication or reduced due to a deletion. So I'll take you through an example to look at all triple-negative breast cancer models by using this TNBC option here and then clicking on search. Currently, we have 31 triple-negative breast cancer models. Importantly, you can sort these models based on what kind of data we have collected over here on the right-hand side. We're going to now look at this model TM00096, for example. On this page, you'll see a summary of the clinical diagnosis along the top. And in addition, there are many sections of data that can be expanded on in these sections here. One of those sections is our gene profiles shown here, as well as the copy number variations that I described earlier. For some tumors, the histology and growth characteristics and standard of care data have been collected and are available here as well. So this particular model was either treated with a control in black or with some of these therapeutics shown here. And you can see the growth repression due to these drugs. This data is being continually collected and added to, and so the database is ever evolving. And finally, in the variant summary section that we described earlier, each model links to our clinical knowledge base, or CKB database. CKB is a dynamic digital resource for interpreting complex cancer genomic profiles in the context of preclinical and clinical trial response data. If we click on this particular gene variant listed here, we will be taken to the CKB molecular profile page on the next slide. So in this example, this is CKB. This particular NRAS Q61K gene variant has been categorized as sensitive or resistant to certain types of therapies. And you can see where the study was linked here under efficacy evidence. And you can go directly to the original publication. To summarize, PDX cancer models offer several advantages over models based on human cancer cell lines. There's tumor fidelity, in which the PDX tumor retains gene expression profile and structure. And because these tumors have only been passaged a few times, PDX tumors are more representative of the tumors present in current cancer patients. In contrast, many established tumor cell lines have been passaged for many, many generations and typically result themselves with very high proliferation rates that can dominate a tissue culture. These highly proliferative cells do not necessarily reflect the characteristics of the tumors from which they are derived. And finally, by displaying characteristics that more accurately reflect the tumors carried in human cancer patients, the JAX PDX models promise to aid in the development of preclinical therapies that are more likely to demonstrate efficacy when transferred to the clinic. The JAX PDX resource will provide invaluable research to help cancer researchers identify and choose the best models in which to test anti-tumorigenic compounds. This webinar was made possible with your support. Jax Mice and Services is dedicated to providing quality resources, flexible solutions, innovative technologies, and unsurpassed expertise and support to accelerate all aspects of your research. Each dollar you spend on Jax Mice and Services supports our shared research goals and sustains valuable mouse models and resources available only from the Jackson Laboratory. Thank you for watching this video. I hope that this presentation has provided valuable information to help you achieve your research goals. If you have any questions about the information shared today, please contact us using the information on this slide. We look forward to supporting you with technical assistance. Thank you.